Welcome to the second part of Autodock Vina tutorial. Here we will check the folders that were installed after installing the Autodock Vina and the MGL Tools software. So under the C drive in the program files, we can see that the MGL Tools folder is there. Now I'm showing this because it is important and we need to copy our PDB files into this MGL Tools folder later on during the docking studies, before the docking studies. Apart from that, there is another folder which is the Scripps Research Institute. In this folder, we will have a Vina folder. Now, this Vina folder is actually important because it has all the different type of files which are required for docking. Now, what I generally do in my case, just for convenience, I take the Vina folder out here and copy it into C drive, which is outside Vina, and it actually helps me when I do the docking, which I can show you later on. And this is optional. In the next part, which is very crucial, is copying the PDB files which we use for docking, like the protein and the ligand files, into the MGL tools folder. This is very important because we need to import the PDB into the Autodock Vina only from the MGL tools folder. So make sure to copy these two files under the MGL2 folder. So once we have copied those two files in the MGL2 folders, which I already have from before, we will proceed in the next part with the docking preparation. For the docking preparation, we will use Open Autodock Vina, which can type ADT, and this Autodock Vina software will open. Basically, it's the Autodock tools which will open. It will take some time to load. Once it is loaded, the interface looks somewhat like this with multiple options which we can use upon our convenience. The first thing we need to do is to import the protein file. So we can read molecule from the file option, and we have to read it from the MGL tools folder. So we'll go again into the program files, then to MGL tools, and then we will go into the protein file, which is the BFA, and we will open it. Now here we can see the structure of BFA that has been imported. It is shown as sticks. You can see it as cartoon forms or ribbon forms, whichever you feel like. But for here, I'm just showing you as sticks. So in the next part, we can do is delete the water molecules. Although I have deleted it earlier, but it's also good practice to delete water molecules once we import any protein into this Autodock Vina software. Now, once we have deleted this water molecule, we'll proceed with the second step, which is adding polar hydrogen. So we will go to this option, we'll add polar only option. For this, we have to tick it and we will click OK. And once we click OK, we can see those white atoms have been added. So we can see additional white colors into the sticks, which signifies that the polar hydrogens are added basically to the polar groups. Now we have to open this configuration file. This is very important. We have to make a configuration file, which is just a text file. You can prepare the configuration file just by looking at the format, which is pretty easy and straightforward. So in the configuration file, we see there is a protein as well as a ligand file. Now we have to save this protein first. In order to do that, we have to go to grid. Under the grid, we have to select macromolecule. We have to choose the macromolecule, which is here, which is the BFA, and we have to select it. And now the important thing is we have to save this file, which is as PDBQT format. Now this PDBQT format is taken up by the Vina for docking studies. So we will go to the Vina folder. Be careful. We are going to the Vina folder and now saving it as .pdbqt, not PDB but PDBQT. I generally try to write as .pdbqt in most of the cases because sometimes it doesn't take it by default. So after this one, we can just save it into this Vina folder, and this will be saved as .pdbqt. Now we have to write the exact name of the protein by which we have saved the protein in the Vina folder. So the name in the text in the configuration file of the protein should be same as the name of the PDBQT file. The next part is about the grid box selection. So we'll select the grid box, and this grid box, the first thing we have to do is to increase the spacing to 1, which I generally do. Now this is done for blind docking, that is we are not specifying any particular region and we are just doing the dock for the entire protein. Now we can play out here by sort of increasing the x dimension, y dimension. The goal would be to increase this box in such a way by clicking this x and y dimension and increasing it and sort of scrolling it on the left and right side. So here we can increase the size of the box and we can increase the y dimension and the z dimension also, just to make sure the residues are all inside the box. So you can just left click on those rectangular boxes given there in red, green and blue, and just scroll it on the left and right side to check to increase the box size. Now the next part is that the center grid box. So this we can actually left click and move it. So this will actually move the box up, down or left and right. It won't increase the size of the box. It will just move box left or right as per the axis. Now once you're satisfied with it, you have to go again to the configuration file, and this is pretty important because you have to write down the values of the center X, center Y and center Z. You have to copy the files as shown here and the exact values of the center X, Y, Z, and along with the center X, Y, Z, you have to also specify the dimension of the box. This is the size of the box. So in the top part of the so in the top part, you can see that it is written in red, blue and green. What are the values which are written? So those are the size of the boxes. So we will enter accordingly. Once we have put everything, we have to make sure to save it as close current saving. So once this is done, we will find out that the protein looks somewhat like this, and the protein is now ready for docking. So once the protein is ready, I will go and sort of hide this molecule by right clicking it and selecting the hide option. The next part, we have to import the ligand. Similarly, we will go to the ligand option like this and go to our MGL tools folder, and MGL tools folder, as we remember that we have saved a PDB of both protein and ligand. So here we have to select the file format which we are looking for as PDB, and we can open this one. Generally, what I see here is the structure of the ligand like this. I generally export it as it is, but you can play with the torsion. Sometimes you can adjust the torsion like this from these options. But I just select the default torsion and save it again as PDBQT under the Vina folder. Once you have saved it, make sure that the names written in the configuration file are exactly the same name by which you have saved the PDBQT file. This applies both for the protein as well as for the ligand. Thank you for watching this video. In the next part, we will see how we can perform the protein ligand docking using Autodock Vina, and how we can analyze the dock structure and also look at the binding sites of the protein.